We are really fortunate today, and I couldn't be more thrilled to introduce Professor Richard Miller. Dr. Miller is a professor of pathology at the University of Michigan, and he's the director of Michigan's Paul F. Glenn Center for Aging Research. After postdoctoral studies at Harvard and Sloan Kettering, he moved to Boston University in 1982, and then to his current position at Michigan in 1990. And he assures me that the weather is much more pleasant than most of us think. He served in a variety of editorial and advisory and leadership positions on behalf of the American Federation of Aging Research, or AFAR, and the National Institute on Aging, and also served as one of the editors-in-chief of Aging Cell. He has received, as you can imagine, many awards for his research, including the Nathan Schock Award, the Allied Signal Award, the Irving Wright Award, and the Kleemeyer Award for Aging Research. He's a senior scholar of the Ellison Medical Foundation and a fellow of the American Association for the Advancement of Science. His research program includes ongoing studies on the mechanisms that link stress and nutrients and hormones to delayed aging in mice, the development of new approaches to slow aging and disease through drugs and other compounds, and early life dietary restriction and the impact of that on aging. He also works on targeted mutations and studies ways in which cells from long-lived birds, rodents, and primates may differ from those of short-lived species. Again, we couldn't be more delighted to have you here, Dr. Miller, and please take it away. The floor is yours.
So I'm really proud and pleased to have been invited to talk to this distinguished group, and also pleased with the notion that our conversation today may be made available to others in the future. My main goal for today's talk is to convince you of something that the last 2 decades have proven, but which most people still don't quite really understand or accept, which is that you can actually slow the aging process. There's a sort of proverbial saying that, there's nothing inevitable except death and taxes, and people consider aging as inevitable. And although it is inevitable, it can nonetheless be delayed and postponed. So my job today is to tell you the data that have convinced me, and I think many other people, a growing number, to whose ranks I'm hoping you will be added soon, that that's the case. And also to show you how the research in this area is working its way towards the time at which it will be possible to develop preventive medicines for people that will let people stay healthy for another decade or two by postponing most of the diseases of aging and stay alive because they're healthy during that period. I'm going to use the term aging today to mean just what you think it means, a process that converts healthy young adults, like graduate students, into less healthy older adults, like tenured professors, with a progressively increasing risk of illness, injury, and death. To me, this makes very good sense. Some people say, oh, my cells are aging, and that's not true from this definition, or this wine is aging. That's not true from this definition. I'm talking about not the aged, not the elderly and what they're like, but about the process that makes the elderly, starting from graduate students and younger adults. The first thing I'd like you to start to think about is that aging cannot really be cured one disease at a time. And here's why. This is the risk rates, the number of deaths per 100,000 people per year in 1997 in the USA. And the main thing to notice is that the y-axis there is logarithmic. Every jump goes up tenfold in the risk rate. In fact, for nearly all the major diseases of human aging, it takes about 8 years for the risk rate of that disease to double. So that if you can imagine a situation in which you suddenly had cured cancer, that orangey line for cancer just went away, it went to zero. The people, as they got older and older and older, would live a little tiny bit longer before they bumped into the next line, which might be heart disease. If you were lucky enough to have a complete cure for cancer and a cure for heart disease, people's lifespan would go up just a little tiny bit, because it would only be another few years before they bumped into stroke and emphysema. In fact, the calculations were done by Jay Olshansky and his colleagues. This is from a paper that they published in Science magazine in 1990. This is the number of years of life that would be left to a white American woman who is 50 years old. That gray bar at the top shows that she would have 31 more years of life. She would die on average at about 81 years of age. And that's still true today. And what Jay and his colleagues calculated is, what if none of those people ever got cancer? They never died of cancer. What would happen? And the answer, when you go through the calculations, is you've brought that woman a surprisingly small benefit. You've brought her 2.7 years of additional lifespan on average. Actually, if you cure heart attacks, you also get about 2.8 extra years. And if you cure cancer and heart attacks, you're buying that woman about 6 years of additional lifespan. Instead of having 31 years to go, she'd have 37 years to go. Now, that red bar at the bottom is the only part that's hypothetical. That's not real data from humans, but that's the data you would see in humans if you could slow aging down using a diet that has been shown to slow aging in rats and mice as far back as 1935. These diets lead on average to a 30 or 40% increase in median lifespan. So you can see we already have in the lab ways of slowing aging in rodents that are 10 times better in terms of additional years of healthy lifespan than you'd get from a complete cure for cancer in people. This is a theme that I'll return to a couple more times. No one really wants to not eat 60% of food, but there are now pills that we can put into the diet of the mice and rats that accomplish the same thing or close to it. And that's what most of my talk today will be about. So it's been known for a long time that aging can be slowed by very low calorie diets or by changes in single genes. Let me show you some examples of that. This is what calorie restriction looks like. The blue dots are a survival curve for regular old mice. This is work done by Andrzej Bartke, but there are now 40 labs that have replicated this. You can see about half of the animals have died by 750 days, and the last regular animal, control animal, dies down at the bottom there at about 900 days. If you give them 30% less food than they want, their aging slows down, and the last mouse actually survives almost out to 1,200 days. This is about a 30% increase in their mean, median, and maximal longevity. Now, these animals are not sick, elderly, arthritic, demented animals. No one wants a pill that will take somebody and allows them to cling to life in a state in which they're not at all happy with their state of living forever. What these animals are healthier. That's why they live so much longer. Caloric restriction slows cancer, it slows arthritis, it slows kidney disease, it slows memory loss, loss of muscle strength, immune decline, and a lot more. And that's the evidence that allows us to conclude safely that this actually slowed the aging process. It's not just pursuing the most common disease or two that kills them. It slows down a very wide range of the things that happen to old people and rats and mice that we don't like. That's how we know it's slowing aging. To give you, I'll show you just one example of that because it's one of my favorite slides. This is work done by Roger McCormick. He took a batch of regular old rats. These are the ones in the black squares down there at the bottom. And he simply put an exercise wheel in their cage. They were curious. You could see that they exercised about 1,000 meters per day, 1 kilometer per day for a few months. But by the time they were 12 months of age, they got bored with running, just like most of us get bored with running, and they never run again. And most of them are actually dead by 36 months of age. But if you put that same exercise wheel into a cage of calorically restricted rats, these are the open circles at the top, they do 6 kilometers a day. And this is not rat adjusted kilometers. This is like 6 real kilometers, the distance from my house to the medical school. And they keep on doing that until ages at which most of the regular old rats have long since died. Even at ages which correspond to humans at the age of 100 years or so, up there at 36, 37, 38 months of age, they're still doing a kilometer every night. So these animals are quite healthy. It's almost certain that that's why they live so long, but it's an important thing to remember. If you think about what an anti-aging intervention might do to you and your friends. When I tell my friends in the humanities, my wife was an English professor, and I stopped telling people that I worked on aging because they had this view of somebody compelling them to become like today's 100 or 110-year-old person and then staying there forever. The data, not just fantasy, these data and others that I'll show you in a moment, suggest that that's just incompatible with the evidence we have. The way in which these anti-aging interventions work is by preventing disease and disability for a very long time. So almost always in a question and answer period, somebody says, hey, would this work in people? And there are two answers to that question, depending on just what you mean. If you ask me if I think whether you forced people to eat 40% less food than they really want, like we do our rats and mice, would it slow aging? And my opinion there's no data on that, but my opinion is, yeah, sure, because it works in dogs, it works in horses, it works in 20 or 30 other species that have been evaluated. It would be, including almost certainly rhesus monkeys. And so it would be very weird if humans were the only mammal on Earth for which caloric restriction did not work. However, sometimes when people ask the question, they're asking, is this a useful approach in the clinic, in public health regimes, to promote slow aging and exceptionally long health span in people? And there, I'm sad to say, the answer is very likely no. If you take a couple hundred people, this is the Aging Institute did this in the calorie trial, and you give them, these are highly motivated, they are not obese, they have a normal range basal metabolic index, and you give them all the support, you give them controlled diets they can pick up every week. They do lose weight. They lose about 10% of their body weight, not the 30 or 40% that is really necessary to get slow aging mice and rats, but they lose 10% of their body weight because they're highly motivated, lots of support, and then they gain it right back. And within 2 years, 95% of them are at the same weight they were at when they started. There are a small number of people. There's a caloric restriction society. It has a membership of 50 or 60 or 70 of people who have the kind of personality, obsessive, very health concerned, that they really can lose 40% of their body weight and keep it off forever. I weigh 180 pounds, so I would have to lose 50 pounds and then keep it off for the rest of my life. And you would too. And people just can't do that. It's like saying people could fly if only they could flap their arms fast enough to take off. People are not built with the behavioral capacity to actually lose 40% of a healthy body weight, keep it off forever. So in my opinion is that caloric restriction is a terrific research tool. We need to learn how it works. We need to know what it does and how what it does is connected with slow aging so that we can develop pills which will have high clinical compliance that do the same thing. The second example I'll show you before I turn to drugs is the Sneldorf mice. The adorable mouse there on my left called Yoda is a Sneldorf. It has a single gene mutation. It differs in only one genetic locus from his sister, Princess Leia over here. And that Sneldorf mutation messes with the pituitary so they don't make growth hormone. And so they're 1/3 of normal body weight throughout their whole lives. And as it happens, they live a lot longer. They live 40% longer than the controls. The gold triangles there, those are the control mice. And the last one dies at a little less than 1,100 days, whereas at that age, 80 or 90% of the Sneldorfs are still alive. The last Sneldorf doesn't die till about 1,450 days of age. And just like the caloric restriction animals, they're not merely alive, they are youthful. That's why they are alive. Cancer is delayed, cataracts are delayed, arthritis is delayed, immune senescence, skin changes, memory loss, and a lot of other things. So the caloric restriction diet and several different gene mutants, I've just shown you one, but there are now 3 or 4 of them that have been replicated, slow aging, postpone a wide range of age-associated diseases and deficits and losses, and thereby allow them to stay healthy and alive for a much longer period of time. Now, the main deal in the dwarf mice is a pair of hormones, growth hormone, which comes from the pituitary, and IGF-1, which comes from the liver, but only when growth hormone is present. And I'm interested, and others are interested, is this like a mouse thing only, or does it apply outside of mice? And there's really strong evidence that it is not just a mouse thing. If you want to go to smaller, less cuddly animals like worms, flies, and yeast, low IGF-1 causes lifespan extension in those too. If you like nice, cuddly, friendly animals like dogs, dog breeds that have very low IGF-1 and growth hormone, the smaller dogs, of course, like the Chihuahua and the West Highland White Terrier and the Cairn Terrier, they are longer lived. They are 40% longer lived than the Mastiffs and the Wolfhounds and the St. Bernards. There are 6 breeds of small horses like the American Miniature Stallion, the Icelandic Pony, the Falabella Stallion in Argentina, and several others, for which there are decent lifespan records. And they too live longer than regular size horses, the kind of horses you see as cowboys running around. There's even really strong data making this connection for cancer in people. There are 6 humongous studies like everyone in Norway or all the doctors and nurses in the United Kingdom, not little tiny studies, which established that short people are protected against essentially every kind of cancer. The exception is smoking-related cancer, but except for smoking, every other kind of cancer, there's roughly a 40 to 50% lower risk than in taller people. It's a huge effect. And the interest for cancer biologists ought to be apparent. Cancer biologists don't apparently think about this or care about it because it didn't come from a cancer study, but it's nonetheless an astonishingly important clue as to ways you might be able to prevent cancer in people. This is some of the dog data. Each dot on that graphic is a breed of dogs. There are a couple of hundred Chihuahuas, a few hundred St. Bernards. One of those dots has my beloved Labrador, probably this one here has my beloved late Labrador Retriever in it. You can see the key number there is highlighted in red. It's 56%. People bred dogs for a long time to get big dogs or little dogs. They wanted big dogs to be scary or little dogs to sit in their lap. They weren't breeding for lifespan, of course, but you can see there's a 56% associations, 56% of the differences amongst dog breeds in their average lifespan is controlled by how big they are. Once I tell you how big the dog is, you know 56% of what you need to know to make a good guess as to how long they're going to live. It's an amazingly strong correlation. Steve Ostad, the guy on the right with the big dog and the little dog, is one of the people who's really been a pioneer in this kind of analysis. So it's dogs and it's also horses. This is an Icelandic pony. It's really small. The guy on the left there is his owner. The name of the horse is Tully. I don't guarantee this is the longest lived horse that ever lived, but I can pretty much guarantee it's the longest lived horse whose picture I could steal from the internet. Tully died apparently of heartbreak a few years after his master died in his 90th year. Okay, so if you're slow in joining us, here are the key points so far, the things I hope you have learned. Aging can be slowed down, despite what we used to think. It can be slowed down, and doing so delays nearly all the late life diseases. So if you would like to have people not get late life diseases, what you really ought to do is slow down their aging. That's what the rest of the talk is about. Now, we're not going to slow aging in people by changing genes in babies. We're not going to take babies and manipulate their pituitaries. Trust me, that's not a grand idea. And heroic caloric restriction diets have very low patient compliance. That's not going to work either. But the genetic mutants and the caloric restriction studies are experimental clues to where we ought to look if we really want to find anti-aging drugs. So we can do it by diets, we can do it to single gene mutants. And 20 years ago, a batch of us got together and said, what we really need is drugs, let's figure it out. So there was a conference, it was organized by Huber Warner and Nancy Nadon, who were professionals at the National Institute on Aging. They invited a batch of us, about 20 people, to help them work out a program for testing biological interventions to promote healthy aging. And that matured into this, the National Institute on Aging's Interventions Testing Program. This is a screen capture of the website made a week ago. The ITP, if you want to know more about it, just search for the ITP on the National Institute on Aging website. You can get a ton of information, including every single paper we've published and every drug we've ever tested and published about. So this is a going concern. We are now in our 18th year. Let me tell you about the program, the ITP program. These are the key features. We use genetically heterogeneous mice. That's a big deal. Almost all science uses one kind of mouse, an inbred mouse called C57 Black 6. That's a terrible idea. Almost everybody does it. 95% of the requests for old mice from the Aging Institute are for that kind of mouse. But then you get information about only one genotype, and it's a sick little mouse. We would never make that mistake. So all of our mice they have the same 4 grandparents, so we can make these genetically heterogeneous mice as often as we want, as many as we want, any year in any lab in the world. But every mouse is genetically unique. They're all brothers and sisters. If I take 2 mice from my colony, I guarantee they share half their genes, exactly half their genes, but it's a random half for any pair. Just like you would share half your genes with your brother or sister, any 2 mice in the colony share half of their genes. Now, this is community science. Anyone, including you, can suggest a drug. The application process, we have an annual application process, and anyone can just say, please test this drug. Here's why I think it's a great idea. Here's why I don't think it will hurt mice. And once we have those applications come in every February, we have a committee that looks through them, evaluates them, and ranks them, and then we pick 5, or if we have a lot of money that year, 6 or 7 to test in our lifespan experiment. The other thing that's expensive, but I think really good, is we test every drug at 3 sites independently. One of the sites is at the University of Texas Health Science Center in San Antonio. That's Randy Strong, the redheaded guy with the beard who's smiling there. And then at the Jackson Labs. David Harrison founded the program at the Jackson Labs, and it has a co-director now, Nadia Rosenthal, who's the science director at the Jackson Laboratories, and my site at Michigan. And the important reason for testing it at 3 sites is that if we see a good, strong result at all 3 sites, we can be very confident that it's replicable elsewhere. If we see a drug working at only 1 site, but not at the other 2, we report it. It's interesting, but we know that we can't lean on it with quite as much confidence as we would be able to exert if it had been given the same result at all 3 sites. And the last point I'll make is statistical power. We use a lot of mice. We wanted to be able to detect even 10% changes in median lifespan for either males or females, pooling the data across site. And if there was a 10% effect, we wanted to see it 80% of the time. We wanted 80% power to detect a 10% change. What that means is that for every drug we decide to test, we have 300 mice getting that drug. 50 males and 50 females at Michigan, 50 males and 50 females in Texas, 50 males and 50 females at the Jackson Lab, and each year double that number of control mice. So that's expensive, but it does give us the power to detect 10% change. 10% doesn't sound like it's a lot, but I'll remind you from that slide I showed you from J.L. Shansky, it's more than twice as large a change as you'd see if you cured cancer in people. So in that context, it is quite worth detecting. Now, most drugs don't work, of course. If I asked you, no matter who you are, how good you are, no matter what you know, to pick a drug that you think is going to slow lifespan, any of us would get that wrong most of the time. Most drugs don't work. And here's the list of the 23 drugs we've published that we tested that didn't work. I've put in red the ones that are most famous. Curcumin, resveratrol, which people are making hundreds of millions of dollars peddling to gullible patients. Fish oil, same comment. Metformin, nicotinamide, riboside. These are the ones which you find at CVS, not for metformin, it's a prescription drug, but many of the others are available as, quote, a health supplement or something like that. And at least in mice, they don't do any good at all. However, the reason I was invited here is that some drugs do work. And this is the first of our publications to show that. The original publication was in 2009 in Nature. This is a replication study published in 2014. So rapamycin, originally isolated from a microbe, has seen some human clinical benefit because it can block immune rejection of transplanted organs. And there are a few rare tumors that it also has therapeutic efficacy. But when you give it to mice, it inhibits aging, it slows aging. And you can see the females, you can get, even at the lowest dose, the green dots, substantial increase in mean and maximum lifespan. The largest dose, 42 parts per million in the diet, has a bigger effect. And it works at the two higher doses in male animals as well. This is what the numbers look like. So the doses in this particular replicate study were between 4.7 parts per million and 42 parts per million. In the males, that highest dose gave a 23% increase in median lifespan. And in the females, the highest dose gave a 26% increase in median lifespan. So each of those is sort of 7 times better than you'd see in people if you had a cure for cancer or a cure for heart attacks. Now, we know what rapamycin does. There's an enzyme in every cell of our bodies and every cell of the mouse bodies called TOR, which conveniently stands for target of rapamycin, and it inhibits that. And knowing that rapamycin slows aging in mice opens the gates to a very large array of studies as to why would it be that inhibiting TOR by a drug slows down brain aging and immune aging and bone aging and muscle aging and cancer and stuff. So once you've got a drug that you know slows aging and postpones all of these forms of aging and all these diseases, that gives you really vital guidance in developing new studies to try to figure out why that should be. That's a central theme of today's talk, which I'll come back to in a bit. So this puts that rapamycin bar into the context of Olshansky's demography. And you can see that if rapamycin worked in people, which we don't know, but if it worked in people to lead to the same kind of lifespan extension that it does in mice, it would be better than cure for cancer, heart attack, stroke, and diabetes in terms of the amount of healthy, active lifespan produced. Now, these rapamycin-treated mice not only are alive, they are in many ways youthful. So here's a study in which we took a batch of mice, put them on rapamycin, and then they were 22 months of age, which is sort of the equivalent of a 50 or 60-year-old person. We euthanized them and we looked at different organs to say, how about aging in the liver? How about aging in the heart? How about aging in the uterus? How about aging in the adrenal? And this slide, which was done by our veterinary pathologist, J. Irby Wilkinson, shows that if you look at the liver degeneration thing up in the upper left-hand here, old mice have got a lot of it unless they've had rapamycin. If you look at the myocardium, there are changes in the nuclei, which old mice have a lot of, unless they've been treated with rapamycin. The same is true for hyperplasia of the uterine endometrium and also for adrenal tumors. Sue Brooks looked at their tendons. This is basically, if you stretch the tendon, how well does it snap back? Old mice are terrible at that. Tendons from old mice are terrible at that unless they've had rapamycin. And this is a different way of studying tendon elasticity at the same time. So tendons don't have cells, right? They're not part of cancer. They are not anything that's going to kill these mice. But the things that aging does, like give you stiff tendons, whatever that is, rapamycin slows it down. And here are 5 more things that Irby looked at, and 4 of those 5 show the same thing. They go up with age in the mice, and rapamycin slows it down. Now, the biggest surprise of the rapamycin studies is the one that's shown on this slide, which is that rapamycin works even if you give it to middle-aged mice. I bet against this. I thought it would work only if you gave it to young, healthy mice. I thought if the mice were already 20 months of age, sort of the equivalent of a 50-year-old person, it would be too late. You wouldn't get any bang for the buck. And I was wrong. Quite pleasantly, giving it in middle-aged mice gives the same effect, just as strong an effect on lifespan in both sexes as giving it starting from 9 months of age in a younger adult. That's very good news because some of us, and that includes members of Congress, are middle-aged, and they really want to know if there's something they could take or give to their friends that would help them from getting older. And they're really interested in drugs that will slow the aging process, even though you're no longer in your 20s and 30s. So the empirical evidence here is good in that sense. We certainly want interventions that can work even in those who are already middle-aged. But in terms of scientific data, it's also really interesting. There is something going on in middle-aged and older mice, which rapamycin blocks, which can lead to a very quick, very speedy change in the risk of mortality. The second drug I'm going to talk about is Acarbose. This is an FDA-approved drug. It's used for diabetics. And what it does is it stays in your GI tract and it blocks the digestion of polysaccharides. Your intestines can't absorb polysaccharides. What they use is bacterial enzymes to turn them into sugars, and then the sugars are absorbed into your bloodstream. Well, Acarbose blocks that enzyme so that when you eat a couple of donuts for breakfast, your sugar does go up, but it goes up much more slowly and it doesn't reach that peak. It slows down the rate at which sugar can be taken up from polysaccharides. And as you can see, it extends lifespan very nicely in males and significantly in females. It's not as strong in females, but it's statistically significant in the females too. This was our first study, and we've repeated this now at 3 different doses of Acarbose, and it always works. It gives an extension in both sexes, and the effect in males is always stronger. We obviously wanted to know, what if we started in middle age? And the answer with Acarbose is that yes, it still works in middle age. It's only about half as good in terms of lifespan extension as if we started it in the younger animals, but it still works. It's not as good as rapamycin, but it's effective even in middle age. And here's that replication study I mentioned to you. We looked at 3 different doses. 1,000 parts per million is the dose that was in the original study, which turns out to have been a pretty lucky guess because that's the optimal dose in both males and females. Now, what about the health of these animals? And I don't have time to walk you through every little bit of this, but let me just summarize it briefly. Mariana Sudogorski, who's now at Wayne State, looked at brain inflammation. She looked at the hypothalamus, and Acarbose, in males only, blocked age-associated brain inflammation. Charlene Day, a cardiologist who's just moved to Penn, Charlene looked at ventricular mass, which goes up with age in mice, just as it does in people. And Acarbose in males, and only in males, blocked that age-associated change in heart anatomy. Mike Garrett, who's just left and set up his own lab in New Zealand, looked at glucose control and found that Acarbose, whether you start in youth or start in older age, improves glucose tolerance. And of course, it has an effect on one of the most interesting health outcomes, which is cancer. It's cancer that kills most of these mice. So this is why we feel comfortable stating that Acarbose has a genuine anti-aging effect. It is blocking age effects in the brain, in the heart, in the glucose control system, and also neoplasia, presumably lots of other things, but I don't have time to go into all of that detail today. The third of the drugs I'll tell you about is 17-alpha estradiol. You've all heard of 17-beta estradiol because it's called estrogen. If somebody says, let's give him some estrogen, or let's give her some estrogen, they're talking about 17-beta estradiol. This is a different chemical in which one of the bonds has been flipped into another orientation, 17-alpha. And for that reason, it's non-feminizing. That is, it has about 10-fold less ability to trigger the famous estrogen receptors, which are so important in, say, breast cancer, among many other things. So our collaborator, Jim Simpkins, said, I'll bet you will create male mice that live as long as female mice without turning them into girls. And let's give them 17-alpha estradiol. So here's what you see. It has a great effect on longevity in the male mice. It has no effect on longevity in female mice. Actually, the male mice treated with the drug live longer than the females, whether they've been drug treated or not. So Jim was wrong about that. It doesn't bring them up to the females. It pushes them way beyond where the females go. We think we have a clue why this is male specific. Oh, before I get to that, let me again show you a little bit of the health stuff. So this is from Mike Garrett. Males, as they get older, their grip strength goes down. But 17-alpha estradiol prevents that, or prevents a lot of it, and in males only. As males get older, their ability to stay on a rotating rod, the rotor rod test, that goes down, unless they've received 17-alpha estradiol, in which case they stay at the original youthful level. And again, that doesn't work in females either. So there's reasonable evidence, which is now growing, that 17-alpha estradiol not only extends lifespan, but slows many aspects of aging. Once again, the hypothalamic inflammation thing is male-specific. The grip and rotor rod I've just showed you. And Mike also, before he left to set up his own lab, looked at a lot of biochemical changes. The most striking of these is elevation of amino acids in the serum. We don't know what it means, but you can see every amino acid except one is elevated by 17-alpha estradiol, and in males only. We need to come to grips now with what that means. And of course, cancer, the thing that kills them, is delayed, but only in males, by 17-alpha estradiol. If you start 17-alpha estradiol in middle-aged mice, we have 3 winners in a row. It works fine if you start in middle-aged mice. And again, it's males only. So we have a clue as to why it might be males only. And this is the clue. It's a collaboration with Mo Jain, who's a metabolomics expert. Mo looked at 2 steroids in the blood of these mice. One of them shown on the left there is estriol 3-sulfate, and the other one is 16-hydroxyestriol 3-sulfate. And you can see that males and females, those are the black dots, don't have much of either of these things. But if you give the males 17-alpha estradiol, that's the blue symbol, it goes way up. This one does, and this one does, like a 20-fold or more increase in these estriol molecules. Females don't do that. So the males, and only the males, have an enzyme, and we know what the enzyme is, that will take this drug, which we put in the food, and turn it into these bioactive sulfated estrogens. So we suspect the obvious hypothesis that this is the good stuff, that 17-alpha estradiol isn't doing any of this. This is the good stuff, and the females just don't make it. So we're doing the obvious experiment. We bought some of this from a store, and we're giving that to male and female mice. What we're hoping for is it will produce lifespan and health span benefits in both sexes. And the last drug I'll mention is this one, canagliflozin. This is an FDA-approved drug. It's a diabetes drug. It inhibits a molecule that is in the kidney called SGLT. Clinically very important, and it also, like Acarbose, prevents increases in blood sugar after you eat a starch-heavy meal. That's why it's good for diabetics. But unlike Acarbose, it doesn't work in the GI tract, it works in the kidney, it has an entirely different locus of operation. And you can see the data, this is published just this year, canagliflozin has a really nice effect, and it is seen only in males. So for reasons we don't understand, blocking those daily glucose peaks is really good for male mice. Whether the effects in humans would be sex-specific or not is an empirical question. We don't know. We also don't know, and really want to find out, how is it that blocking these daily glucose peaks after a carbohydrate meal slow down virtually all forms of cancer? We would love to know how we can get this to work in females for reasons that are obvious, not just in terms of scientific interest, but in terms of the clinical translation. And we also now need to turn to our epidemiological colleagues, because there are an awful lot of people who are taking canagliflozin and other drugs in this class, often for diabetes, but it's possible that they would work in non-diabetics too, to accomplish health benefits. We need to think about clinical trials and outcome there. Now, I won't talk to you about the other winners that came from the ITP program. I'll mention them just in passing, but the effect is somewhat smaller, so we're not really studying them. One of them is an antioxidant called NDGA. One of them is an over-the-counter health additive called Protandim. Gave a small effect, and it was in males only, but it was there. One of them is the amino acid glycine. Here, it worked in both sexes, which is nice, but the effect was really small, 5%. And so we're not pursuing that. Often people want to know, what drugs are you now testing? And these are not secrets. These are on the ITP website. You can find it if you do a search for ITP and compounds. But I'll list them here for you so you can see what is currently in the pipeline. Every year in my lab, we have several hundred mice that are in their first year, several hundred mice that are in their second year, several hundred mice that are in their third year. The ones that are in red are special, and I'll show you just a little bit about each of those, because those are the ones that look like they're working. We can't publish it yet. We have to wait till we have more data, but we're allowed to talk about it. And so I'll talk about it. These are the ones that in unpublished preliminary data appear to be most promising. And I'll mention that astaxanthin and meclizine are available at CVS. They are non-prescription. They are alleged to be beneficial. Astaxanthin is alleged to be a beneficial health supplement, and meclizine is Dramamine. It's good for you if you're going to get seasick or have air sickness. It blocks motion sickness, so it's available without a prescription. Captopril is a famous high blood pressure drug. So let me show you the unpublished data for meclizine, and you can see why we haven't published yet. We want to wait another 6 or 8 months for more of the mice to die, but 57% of our males are dead, and you can see meclizine, the red line, already looks pretty darn good, and there's no indication that it is helpful for females. There are 2 important caveats. None of these mice got old yet, so we don't have any information yet on late life deaths. We really need to know if this is going to continue to work. And especially for the females where we have fewer deaths, the data are far from complete. And this is the other unpublished data I'll show you. This is astaxanthin. And again, it looks really promising for males, even for females. There's a mistake here on the slide. It says males, but it should say females. Even for females, it appears to have a hint of promising results. We really won't know for another 6 to 10 months whether astaxanthin is giving a significant effect in either sex, but certainly the female data set is incomplete. So this finishes the sort of database part of my talk, and I've summarized here the sort of percentage increase. Anything in red in that column has a significant effect in that sex. So we have 6 drugs published with significant lifespan benefits in male mice and 3 significant lifespan benefits in female mice. This column here shows whether they've been replicated or not. The older ones have all been replicated. The newer ones haven't been, but are going to be. The metaphor down here is this is what tools looked like a long time ago. This is what choppers looked like in Paleolithic times. That's about where we are now. We have tools that can slow the aging process. What we want is this. This is a Swiss Army knife. And we have refined tools that are sharper, more pointed, able to do what you want when you want them, and are designed for various purposes. One of the major points of research in anti-aging pharmaceuticals is first to prove that these exist. We've got that. I think I've convinced you of that. And now to move to this area of refinement. So this slide talks about why the ITP is important, taking the long view. I've mentioned this before, and I'll mention it again because it really is important. Aging can be slowed, and in mammals like us, not just trivial little fruit flies and worms and stuff. It also gives us new targets for intervention. mTOR, the target of rapamycin, the peak glucose levels that are addressed by canagliflozin and Acarbose, sex-specific steroid targets, as in the 17-alpha estradiol, and maybe targets of astaxanthin and meclizine. Once you have a target, you can bring to bear the whole very powerful set of tools related to modern pharmacology and start to get better drugs that are more potent, that have fewer side effects, decide whether giving them in an on-off regimen is a smart move. All of that can be done, but you need targets to start with. And these are hints for human clinical studies. Even if I were a doctor, which I'm not, I would never say, yes, thou shalt go out and get this drug and start taking it. Promise you it's going to be good for you. We're nowhere near there yet. But the data that we do have at this point provide hints for epidemiological trials, which can be done now, and clinical trials, intervention trials, which are likely to be doable soon. One of them at least is underway. So you yourself can propose new drugs to test. If you have a drug you're interested in, send us an application. If you need help with that, ask any of us for help. About half of the applications that come in were refined by a conversation with me or David or Nadia or Randy. We exist to help you work out applications for drugs of interest. If you do experimental work, you can get these drugs. They're on the open market, almost all of them. You can test it in your own system. If you have a specific interest, for instance, in muscle or bone or brain, we can give you tissues from our freezers full of tissues from these drug-treated mice. If you have something that can be looked at under a microscope, call us. We can give you fixed and frozen sections from these tissues. Okay, that's the data. Let's go into the speculation. What if we figured out how these genes and diets worked, and then we use this information to slow down human aging? As I've mentioned, I don't think we're going to spend any time usefully talking about gene therapy, genetic alterations before birth or of adults, because we don't know what genes to change. We don't know how to change them. We don't know what side effects they would have. Let's just not talk about that, or caloric restriction for reasons I've emphasized. So journalists go to aging meetings and they try to ask everybody the same question: how long could we live? And then the person who gives the biggest number is the person they put on the cover of their magazine. And this is what I call the Fantasyland approach. This slide unites four people who embody that approach: Yoda, who is an exceptionally long-lived alien; Zeus, who is immortal; Methuselah, on questionable authority, is alleged to have lived many, many hundreds of years; and Aubrey de Grey, who has developed a famous program called SENSE, the Strategy for Engineered Negligible Senescence, which Aubrey promises us will stop people from getting frailer and more prone to life-threatening diseases as they get older indefinitely, even at ages many times what we reach today, and moreover will restore the already frail to youthful vitality. People willing to say that's the kind of stuff get famous and deserve as much scientific credibility as Yoda and Zeus and Methuselah. I prefer something called the reality-based approach. We notice that the experimental evidence says caloric restriction in many species can extend lifespan by 30 to 45%. Small dogs live 40% longer than large dogs. Another diet I didn't tell you about can extend rat lifespan by 30 to 42%. The mutants I did show you a little bit can give you a 40 to 60% increase in lifespan. And another mutant I didn't talk about can get you 28%. And so evidence, defensible evidence-based estimate is about 40%. That's not a pledge that we can extend human lifespan by 40%, but it is evidence that it's not ridiculous to talk about lifespan extension in people of about 40%. This would lead to an average age at death of about 112. Some would live longer, many would live half, would live shorter than that. But anything above this currently is sort of wish fulfillment, science fiction, and speculation. And it wouldn't mean, yeah, you'd live forever. You'd not be immortal, of course. What it would mean is postponement of the late life disabilities, including lethal illness. And I'm not just making that up, because that's what we see in the dwarf mice, in calorie restriction, methionine restriction in the small dogs, and in a batch of species I didn't focus on in today's conversation. But evolutionists figure this out. Evolutionists made long-lived critters a lot, and that's exactly what you see: postponement of late life disabilities and lethal illnesses. Now I'm going to make a political talk. The obstacles to finding a way to slow human aging are not my fault. They're the fault of everybody else. 85% of the obstacles are political, and only 15% are scientific. This is what grant funding looks like. The gold bar there is the amount of money that goes to the National Aging Institute. But that's misleading, because half of that is Alzheimer's, and of the remaining half, 2/3 are for how best to take care of old people and social science. The amount of money in the NIH budget that actually goes into the stuff I'm talking about, that is fundamental research into the biology of aging, is too small to be seen on screen resolution with an Excel graphic. I've cheated. I've amplified it enormously, so you'll get the impression that there is some non-zero number. But it's really tiny. The last time I did the calculation, for every $100 spent by NIH, 6 cents went into aging research, the fundamental stuff I'm talking about today. I feel that's a terrible decision, a terrible mistake, because our stuff actually works and their stuff doesn't work. And our stuff has real potential to improve human health. So why hasn't this worked? There are 10 reasons. I'll go through them very quickly. That Milbank Quarterly article that I think was made available to people goes into this in more detail. But the first reason is that most public gerontologists are crackpots, and no one wants to hang out with that sort of person. This is an example. When Deepak Chopra writes a book, it's the #1 bestseller. It sells millions of copies, apparently in hardcover. When I write an article in the Journal of Gerontology or Aging Cell, I'm lucky if 100 people actually read the thing. And so when you say to someone who's not in the sciences or not into medical research, what are aging researchers like? They get this, and they get Aubrey de Grey, and they get others who appear on Oprah, but who don't really have a good grip of the science. I had a really pleasant conversation yesterday with 2 producers for 60 minutes. They want to think about a nice part of 60 minutes on aging research. And like most journalists, they are torn. They want it to be rational, good, solid science, and they want it to be spectacular. They want someone like Aubrey or Deepak Chopra or someone who's willing to make newsworthy claims. And I tried my best to sort of meet that wish fulfillment without actually misleading them. We'll see how it goes. So reason #2 is that aging is viewed incorrectly as inevitable. I spent most of my talk today trying to convince you how wrong that is. But if you are the kind of person, as most people are, who say, aging, yeah, you can't do anything about aging. Once you think that way, then there's no point in actually worrying about how you might slow the aging process down, because your baseline assumption is that nothing could ever do that. We need to beat that. We need to get the public to understand that's wrong. The third point is that voters' relatives die of diseases, and diseases have lobbies. There's a huge lobby in Washington for Alzheimer's research, and another for cancer research, and another for AIDS research. Even fairly rare diseases have nice big lobbies, and politicians listen to them. There is a lobby in Washington that argues for aging research, and there are 3 people, and I know them all, and they're good people, and they're doing their very best, but they're outnumbered 10,000 to 1 by other lobbyists. We need the public at large, but also scientific administrators, to understand that if they really want to conquer cancer and conquer Alzheimer's and conquer diabetes, they should do it the smart way, which is to work on the aging process that underlines all of them. The next 2 points are economic. Drugs that actually slow aging cannot produce quick profits for a drug company. The CEO of I apologize if anybody in the audience is a CEO of a drug company. You might want to mute your speakers for about 30 seconds here. These people want to make money for their company. And by gosh, they want to make money in the next 3 to 5 years. And they want their stock options to be valuable, and they want the stock price to go up. And they need a blockbuster drug before the patent expires on the last blockbuster drug. And they have zero interest in a 20-year test of a drug that actually slows aging in people. It's expensive, it might not work, and they will be retired by that time. So drug companies have zero incentives to actually study drugs that work. However, good news for the drug companies, they make a ton of money selling stuff that pretends to slow aging and produces quick profits, even though it doesn't work at all. I've shown some colorful vials at the bottom, one for melatonin, one for royal bee jelly, one for DHEA, one for somatotropin or something, and then the current poster child, resveratrol. None of these, there's no evidence for any of these that it's good for people. But the FDA rules have been written in such a way that you can, as long as you don't stare into the camera and say, see, this will cure a disease, you can wink and say, we'll keep you healthy longer or something. You can sort of give people the impression that we all know it slows aging, but I'm not allowed to say it because of those bureaucrats. And this works quite well, and it's a multi-billion-dollar business. And because of 4 and 5, we're not going to get a lot of help from the drug companies. Now, a politician, reason number 6, a politician who wants to conquer cancer is a hero. A politician who promises to slow aging is considered a nut. 3 of the last 4 presidents knew this. They, at some point in their presidency, most recently Joe Biden, made a nice speech. They said, we will have a moonshot project. We're going to cure cancer. Nixon did it, and I think Obama did it. No, Obama was against Alzheimer's. One of the other earlier presidents was also anti-cancer, and their ratings go up a little tiny bit. But if you can imagine a president making a primetime address saying, I'm devoting the full power of my administration to slowing the aging process, he or potentially she would be considered crackpot. They've gone over the deep end. They've lost everything. And that's what we've got to fix, because there is no way on the horizon to cure cancer. We can't cure cancer, all cancers, or even many cancers in mice or people. We don't have a systematic way of slowing down all cancers or even the major cancers, despite the enormous numbers of billions of dollars that have been thrown at this problem. But we can do it in one way in the lab, and that is to slow aging. We've got drugs that can do it. We've got diets that can do it. We have single gene mutations. So if you actually do want to protect people from cancer and, oh, by the way, everything else that goes wrong with aging, slowing aging is the way to do it. We already have proof of principle in mammals that that can work. We want to improve public understanding so that you're not considered a nut if you say, I'm interested in slowing aging. Reason number 7, scientists follow money. Young scientists want jobs. There's an enormous amount of money for cancer, AIDS, Alzheimer's. There's very little money for aging, basic aging research. Young scientists, the smart ones, understand this. They want to go into something where they can get a job. And then the next two are linked. There's Potemkin science. That is, there are an awful lot of bad, shiny ideas and a lot of rich people who can't tell the difference between good science and bad science. If somebody with a really good sales pitch goes to them and says, I've got a drug that will stop senescence in its track, they tend not to go to scientists who are skeptical of that claim and ask for a frank exchange of views. They tend to think that they're successful at guessing who's telling the truth and who's not. Now, they're not that good at it by and large, but they don't know that. And so bad, shiny ideas attract an awful lot of attention from venture capitalists. You all know at least 3 or 4 humongous companies founded by people who were good at what they did and now think that as a sideline, they're going to cure aging by hiring a worm biologist or by hiring someone who's really keen on blood transfusions, and they will direct the fortunes of the companies and they will get it right. And as you can tell, I'm not convinced that will work well. The last idea, the final idea of today's talk is gerontologophobia. It's a noun I invented. It's a syndrome characterized by a fear of what anti-aging medicines might do to society if they were ever invented. This is a very personal peeve of mine. My wife was an English professor, and for many years, whenever we had meals with English professors and history professors and philosophy professors, I learned not to tell them what I did, because if I told them what I did, I was destined for 2 hours of you are a bad person. We do not want the society to fill up with old people. So here, for example, this is a sort of a crystallization of that viewpoint. It's an editorial written in 1999 when they were, for the first time, some genes that slowed aging in worms. Thomas Lynch, who is an undertaker poet. He's both simultaneously the best poet among American undertakers and the best undertaker amongst American poets. He wrote an editorial for the New York Times. I'll quote you the sort of money quote here. How far could we go? Too far is one possible answer. Like drunks with drink, enough is never going to be enough. To the medicos, maybe thanks, but no thanks is the thing we should say. Let's leave well enough alone. And I call that position Lynchism. And to summarize it, it's we should stop research on aging because we don't want the world to fill up with old people. Now, if you would accept that, which of course I don't, you would extend it very logically, because that same line of reasoning says, yeah, well, also, we should stop research on cancer and diabetes and stroke, because if we make progress there, we're going to help people stay healthy longer. If you find a way to cure cancer or diabetes or stroke or prevent any of them, the effect is you're going to have more people surviving in a healthy fashion into old age. That's what aging research is designed to do. So if you actually bought the silly logic of the Lynch position, you would definitely want to campaign to stop research on cancer and diabetes and stroke. Actually, if you go back a couple hundred years, you take the Lynch position, you would say, well, kind of wish we hadn't done all that research work on influenza vaccines, insulin, seatbelt, high-yield wheat, penicillin, mammography, etc., because the world is now filling up with too many old, and for that matter, too many middle-aged people. We should go back 200 years to that blessed state where people were dying in their 50s. Now, I'm going to give you a 3-point quiz that will allow you to determine whether you personally are a gerontologophobic, and I'll make it easier for you by giving you all the answers so you won't feel under much pressure. Here's the first question in the quiz. Is it good or bad to save lives by breast cancer research? Good is the correct answer. Saving lives by AIDS research, kidney transplants, making synthetic insulin available to diabetics, that's all good. But saving lives by anti-aging research is bad. That's the correct answer. And give yourself 1 point if you selected that answer. The second question is multiple choice, and it's about population pressures. We should best face population pressures by which of these? You can provide birth control equipment and information. You can use conservation methods to reduce resource depletion, or aim for more equitable resource distribution. Or we could stop all medical research and treatment. All of those clearly are incorrect. The correct answer is we should stop aging research before the world fills up with sick old people not doing their fair share of the work. So if you circled answer E, you give yourself a 2nd point. And the last question is a multiple choice question. It's about ethics. Interventions that preserve health, such as exercise, green vegetables, antibiotics, and appendectomies. The question is here, when are they ethical? And the answer is when anybody uses them. They're ethical when they're used by you, by me, by my mom, by anybody. No ethical questions arise. But if you add anti-aging pills to that list, then the answer changes. The answer is me only. Everybody wants to take that anti-aging pill. If I have a jar on my desk here of anti-aging pills and I offered to give it free to anyone who asks for it, you will ask for it. Okay, but you'd feel kind of bad. So you'd want to give it to your best friends and to your relatives. So it's okay by used by me. It's okay by used by you too, but not everybody, of course. That would be unethical because we wouldn't want the world to fill up with healthy old people, would we? So you can tell now where you score on the gerontologophobia quiz. So my last 2 slides, making lots of grouchy old people is not the goal of anti-aging research. We do not want a lot of people who resemble today's 100-year-olds. We don't want them to fill up our society. And fortunately, there's no evidence that any of these anti-aging interventions actually do that. This is what we want. The real goal of aging research is, this is what a normal average person looks like at the age of 70. That's on Harrison Ford's 70th birthday. We would like a normal person on average at the age of 114 to look pretty much this way. And this is not just a fantasy, a science fiction, a wish fulfillment thing, because the drugs, as well as the diets and genes that I've been talking about, that's what they do. They postpone the bad aspects of aging rather than merely put off death. So I've tried to emphasize, when I've presented original work, the work done by the postdocs in my lab, who are listed at the upper left. We have a heroic team of mouse caretakers listed at the lower left there, and Bill and Melissa do the cell culture work. I've given credit deservedly to Dave Harrison, Randy Strong, Nancy Nadon, Francesca Macchiarini, Nadia Rosenthal, and Huber Warner, who were instrumental in getting the ITP started. Up until 7 years ago, every dime of this was paid for by the National Institute on Aging. And for the last 7 years, we've enjoyed the blessing of funding from the Glenn Foundation for Medical Research.
Well, it's very generous of you, and be careful what you ask for, because you can be sure we'll be coming to you for an update next year after you run more compounds through your platform. So once again, on behalf of the Atria Institute and all of us here today, thank you so much. And we look forward to more interactions and more discussion.